the more exposure you get to these muscles, hopefully it's starting to look a little different. If you're on the front side of the arm, it's going to be a flexor, back side of the arm, extensor. We went over some things last time in the lecture. Hopefully cross over to the left. To finish this up, when it comes to skeletal muscles, prime mover, the term prime mover, sometimes they use this in textbooks to talk about the muscle, the skeletal muscle that produces the desired action. For example, to flex my forearm, I'm going to use a prime mover, which would be the bicep brachii. To flex it, that's my prime mover. But some textbooks refer to prime mover using the word agonist. So these two words can be used synonymously, and it refers to using a certain skeletal muscle to produce a desired action. So prime mover and agonist can be used interchangeably. However, the next term down, antagonist, would be like if I go back to that same muscle, to produce flexion at the forearm, if I use that biceps brachii, that's my agonist. The opposite side of the body is going to have to relax. So the muscle that's opposing that will have to relax, and that's the antagonistic muscle. The antagonistic muscle of flexion would be my triceps brachii. So to get the forearm back into anatomical position, I would have to use my antagonist muscle, which would then become the prime mover to extend the forearm. So you can think of these as one is opposite the other. Agonist produces the desired action, and the antagonist is going to be the opposing muscle that has to relax at the same time. Synergist. A synergist is going to be a muscle that works with another muscle for the prime motion to happen. For example, if you want a nice wrist flexion, you want a nice right down the middle wrist flexion, and you're probably not going to use just one muscle, you're going to use a combo of muscles. For example, you'll probably use the flexor carpi radialis and the flexor carpi ulnaris. Together will give you a nice flexion right down the middle. So those two work as synergists to produce an action where two actions together give you the desired. They work together in synergy. Synergist. Yeah.
Would it only be two synergists that work together, or could you have a synergist working with a prime mover? Or would a prime mover just...
Usually you don't, usually when you talk about synergy, you talk about two working together, and neither one of them is going to outpower the other, so neither one is going to be the prime mover in that case.
Okay.
Yeah. Now a fixator is going to be, well, if I want to flex my entire arm, but then I want to go and have my prime mover at this position. So if I go to this, flex my arm forward, the entire arm, and then if I want to do the same bicep brachii prime mover, I want to hold this arm in this position and then allow flexion to happen at the forearm. But in order for me to maintain this position with flexion at the shoulder, flexion of the entire arm, I've got to fix this in place. Then I can send the signal down by my brain to flex the bicep, which would be the prime mover for my desired action. But I'm not giving up right here. I'm fixating my arm in this position, then I'm going to flex right here. So a fixator in this case, now fixators can be all kinds of things. I can't just say the bicep brachii is a fixator. A fixator fixes a body position in a certain place and allows other prime movers to take over. So you're just simply fixating a place, a certain position, and then you're allowing other muscles to work. So, that's what a fixator is. But you're now talking about more complex motions, more complex actions. You start involving fixators. So a fixator puts a body position in a particular area, and now other muscles can now act to fixate. Okay? Now, what today is going to be about is, how does this all happen? Now, you got some background on muscles now. You know the general idea. Now, if I kind of focus in on one muscle cell taken from skeletal muscle tissue, how does contraction actually occur? Contraction has to happen from the cellular level. So I've got to go into the cell for this to happen. So I'm going to take one cell out of the skeletal muscle tissue. You know that some of the properties, we went over these before, it's striated, it's under voluntary control, multinucleated. Fine. So if I really zoom in here, I got to get into the cell, and I got to get in there and see what is it about this striation pattern, or this striping, that makes it unique. There's got to be something about this because nervous tissue doesn't contract, and it doesn't have striations. Epithelial tissue doesn't have dark down its length inside those cells. Epithelial tissue doesn't have striations. So there's got to be something about the striations, but then you say, so well, how about smooth muscle? Smooth muscle contracts, but it doesn't have striations. Yes, and I'll have to come back to that. But for now, we'll have to look inside a skeletal muscle cell and kind of draw some comparisons with cardiac muscle tissue, because cardiac is striated also. So I got to go into the cell. We got to go into the cell and look at what are these all these striations based on, and what allows this muscle to contract, shorten. So I'm going to go in here, but kind of as a review from last time, I'm going to zoom in. I'm going to have to telescope to this level. So what I mean by this is we've gotten this far, where you have an arm. I'm using a bicep here as like an example, but you could use any muscle, belly. So you've got this origin, insertion, and there's the muscle belly, and I'm pulling out a piece of this gross bicep brachia. If I pull out a chunk of that and blow it up, we went through the covering, the bundles, and then I telescope out one of those bundles. Those bundles are in fact bundling other things. And this right here is one particular skeletal muscle cell. Now remember, skeletal muscle cells are really long. So there's a lot of these muscle cells in this bundle. So what I'm going to do here is, let's label a couple things. This is just the gross anatomy of the muscle. I telescoped out one bundle, so I'm going to say this is a bundle. We went over this last time. That's a bundle. In that bundle, there's a bunch of cells. I telescoped out one of the cells. So I'm going to say this is a muscle cell. A skeletal muscle cell. Okay. You saw this, a group of cells under the microscope is a tissue. You saw this under the microscope. You laughed back. You see the light, dark banding pattern, which is what I was talking about with this. The light, dark banding pattern. So you start to see it at the cellular level. So you got to go into the cell and see what's going on there. Now, muscle cells are really long. Okay? They can run the length of a muscle. So from now on, if I refer to a muscle cell as a fiber, it's the same thing. A muscle fiber, a muscle cell is the same thing. When you start talking fiber, you're talking long in appearance. Long cell. A cell that's just been stretched out, elongated. Skeletal muscle cells can sometimes be called skeletal muscle fibers. So this can be a fiber or a cell. Doesn't matter. You see the nucleus right there, you see the nucleus right there. Multinucleate. Cool. I've got to now go into the cell and see what is making this cell contract, shorten. So now I've got to telescope even more. So I'm going to telescope a little bit more, and I see that the cell itself is packed with certain elements here. So this right here, this that's telescoped out of a muscle cell, a single muscle cell, has a bunch of other things grouped in it. Those things are called myofibrils. Myo means muscle, and then a really fine fiber is going to be a myofibril. So I just telescope out of one single muscle cell, one myofibril. Alright? It's hard to kind of grasp how we're zooming in. Okay, so we're going from large muscle to bundle to cell, and now pulling out of the cell, we're pulling out this very, very thin, very, very fine myofibril. We're now at this, and you really start to pick up this light, dark banding pattern, or striation pattern, when we get to the myofibrilla. We are inside a cell. So then, it really comes down to the proteins that make up the myofibril. Proteins. What are the building blocks of proteins? Amino acids. You put amino acids together based on the DNA code. You put them together by going through transcription and translation. And you bond them together, peptide bonds. So, there are a few proteins that make up this myofibril that we got to list. The proteins involved to get a muscle to contract, you got to know these. The major muscles, or the major muscle proteins that you'll find in this myofibril are as follows. There's four major. Being telescoped out of this myofibril, there's a couple, we'll draw these later. But I want you to at least identify the muscles involved in muscle contraction. The muscle proteins involved in muscle contraction. The first one is going to be called actin. Now I know you've seen actin before. I mentioned actin way back when we talked about cell stuff. We talked about cytoskeletal elements, and some of those microfilaments are called actin, protein. There's another one called myosin. Actin. Lectin is a protein. It's unique. I'll show you the structure in just a minute. Myosin is another protein involved in muscle contraction. Then there's one called, now there's a couple ways of pronouncing that one. I say troponin. I've heard it pronounced troponin. I don't really care. That's how you spell it. Troponin, troponin, it's one of the muscles involved in muscle contraction. One of the major proteins. One of the major proteins in muscle to make it contract. And one last one, tropomyosin. One word. Those are the four major proteins involved in muscle contraction, but you got to get down to intracellular. You got to get into the cell to see where these proteins are arranged. Okay. These are the big ones. I'm going to draw them the way they're set up inside of a myofibril, making up a myofibril. So the next place I'm going to go is I'm going to draw the arrangement of these proteins inside of this. Okay. So, next step. The way this is set up is like so. Alright, first, let's draw. Draw a jagged line. That is not any one of those proteins I named yet. You'll see how the anatomy of a contractile element, of a myofibril, is set up. We'll draw this. I'll draw another one, let's say, right here. Now, to put a label to some of this. This right here is called a Z-line. It makes sense. Looks like a jagged Z. This right here is a Z-line. In your textbook, I don't know why, but they call it a Z-disc. I think it looks like a line, so we'll call it a line. Z-line, Z-disc, same thing. Okay. Now, those four proteins are going to be found in a particular arrangement between the two Z-lines. So everything between these two Z-lines is going to be called the sarcomere. So I'm going to write that on here. Between this point and this point is called the sarcomere. The area between two Z-lines within a myofibril, within a muscle cell, is called the sarcomere. And this is truly the unit that will go through shortening or contraction. So, this will eventually shorten. But I've got to show you the arrangement of how these actin, myosin, troponin, tropomyosin proteins are in this sarcomere. So I'll start out. Let's draw actin first. Actin looks like this. It's made out of amino acids. So I'm going to draw an actin protein like this. Actin, 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 actin. Good enough. Actin is attached to the Z-line. Amino acid to amino acid to amino acid to amino acid. So this is one thin strand of actin protein. But in muscle, you're going to have two of these linked together. So it'll be like this. That's the way actin is set up. It's attached to one Z-line. I'll draw another one. They do not meet in the middle. There is a gap. So one actin protein extends about this far into the sarcomere. The other one is attached to the other Z-line and extends about as far, but they are not attached to one another within a sarcomere. Enough? Now, there are several actin proteins attached to the Z-line. So another one would be like this. Now I'm going to get kind of sloppy about this, but